Chapter 4 is about enzymes and cell respiration, the two main ideas there, enzymes and energy. And cell respiration is that series of processes that are all about us getting energy from food and converting that into cellular energy, the energy that we can use to drive cellular processes. So let's take a quick run through some of these slides. And as usual in the video lecture format like this, you should take advantage of the format and pause the video whenever you need to write down some notes, to jot down some questions, to do a little extra reading in the textbook, or to find another resource if you need to help answer a question. Again, pause the video if you need to, but we're going to go through these first few slides pretty quickly because most of you seem to have a pretty good understanding about enzymes and their importance. We talked about in class how these graphs on the top here showing the distribution of the different energies, the different kinetic energies of molecules in a sample of matter, explained why you get an increase in the rate of a reaction when an enzyme is used. That enzyme lowers the activation energy, and it means that at a given temperature, at a given average kinetic energy, you're going to have a lot more molecules that have sufficient energy to react. And the two models for enzymes, really there's some truth to both of them. The lock and key suggesting the very high specificity between enzymes and many of their substrate molecules, but also it's not a rigid structure like a lock and a key. There is an interaction between the molecules in the reaction, the substrate molecules, and the enzyme itself. And the enzyme and the molecules both change during the process. The enzyme bounces back to its original conformation, and the substrate molecules are typically converted into something new. And we also talked about the influence of temperature on the rates of enzyme-catalyzed chemical reactions. And I'm not going to say anything more than what I've already typed up here. You could pause the video and read these notes if you need to for a review. Similarly, pH can influence the rate of a chemical reaction, and it does so by influencing the shape or the conformation of the enzyme. The enzymes, to function well, have to have a very precise shape, a very precise conformation, so that the enzyme can bind with the substrate molecule or molecules. And so enzymes are adapted to have the correct shape, that precise, exact, perfect shape in the environment where the enzyme normally does its job. So this is showing you some enzymes and the pH optimum for each enzyme. And again, it depends where that enzyme tends to work in the body, and the pH range is adapted for the conditions under which that enzyme normally works. And I don't expect you to memorize all of those, but just to give you an idea. Cofactors are things that for certain enzymes must be bound for them to be active. So this is showing you in general an enzyme in part A that has not bound its cofactor, and so the substrates are not bound. Once the cofactor is bound, the enzyme goes through a conformational change, and now it can bind the substrates and do its job. If the molecule that binds to an enzyme prevents it from attracting and attaching its substrates, we would call that an inhibitor instead of a cofactor. This is another graph that's been a part of conversations in my biology and physiology classes for a long time. I think it's an important idea. It's showing that as we increase the substrate concentration, the reactants of whatever enzyme catalyzed chemical reaction we're talking about, as we increase the concentration of those molecules, the reaction rate increases. But we reach a point where adding more substrate does not increase the rate of the reaction. So in the far right-hand part of this graph, we're adding more of the substrate molecules, but the rate of the reaction doesn't increase anymore. Straight and branched metabolic pathways. Metabolic pathways are a critical part of the biology of all living things. This is the way we break down the stuff that we don't need anymore, and this is the way we build up the things that we need to build our cells. This is showing feedback inhibition of a metabolic pathway. So we have a branched pathway starting with substance C there. And these two different enzymes can convert that substance into two different products. And we can sort of control that branch point in the pathway by feedback inhibition. Let's say we have all of the product here, F prime, that we need. If there's excess F prime building up, feedback inhibition works on key enzymes in metabolic pathways, virtually every metabolic pathway. So we have this enzyme number 3 prime that is driving the production of this molecule F prime at the end of this branch point. If we have lots of that stuff, that F prime can feed back, and it can typically bind to, or something that's related to F prime can bind to this enzyme 3 and inhibit it. It can slow it down or even stop it altogether. And so now all of this product C that is being produced in the earlier stages of the metabolic pathway is going to be used by the other enzyme to drive the other pathway, maybe until it catches up. Inborn errors of metabolism. These are inherited diseases. An error of metabolism is when the genetic mutation, the genetic abnormality, causes an enzyme to be produced that doesn't function the way it's supposed to. It may not function at all. And so if we have an enzyme in a metabolic pathway that is not working, then the product of that pathway isn't going to get made, or perhaps not enough of it is going to be made. And that can lead to a disease state. And this is showing a pathway that is a branched pathway with lots of different enzymes. And when different mutations lead to different ones of these enzymes not functioning properly, we can wind up with a lot of different medical conditions, different diseases. And again, I'm not going to ask you to memorize any of these. You should be familiar with the fact, though, that a lot of inherited diseases are the result of a mutation in a gene that codes for an enzyme. And so that enzyme, as part of a metabolic pathway, isn't able to do its job, and therefore the pathway breaks down, doesn't get accomplished what it was meant to do. And here's a table with tons of these inborn errors of metabolism. Again, you don't need to memorize any of that. It's just to give you a sense that there are lots of inherited genetic diseases that are caused by this type of a mistake. All right, going to wrap up really quickly talking about some energy metabolism. I think most of you are familiar with photosynthesis. Photosynthesis is a process where plants, with a little bit of chemical magic, can convert very low energy molecules, carbon dioxide and water, into energy-rich organic molecules like glucose. Plants can make just about any organic molecule from scratch. We talk about photosynthesis as being all about the production of glucose, and clearly plants make a lot of glucose because they use that as a form of energy, and they also use that to build their cell walls. But they can make a lot of other organic. And going the other direction, plants and animals like us can take those energy-rich molecules like glucose, and we can break them back down into the carbon dioxide and water that they were built up with. And if it required energy to build up those organic molecules, well, it releases energy when we break them down. And living things do that step by step in some long and sometimes complex metabolic pathways. And we wind up with CO2 and water as the end products, the waste products, and a bunch of energy is released that the organism can use to drive cellular processes. You can also get the same energy released by burning that organic substance. We can burn organic molecules, as you are probably aware, and release all of that energy in a flash of heat and light. Well, it's the same amount of energy and it's the same products, but we'd rather release that incrementally so we can use it. And these are some gear graphs that the authors of your textbook thought were good for thinking about, making more concrete the idea that exergonic reactions are needed to drive endergonic processes. And a lot of the processes that living things have to do require energy. And to provide the energy for those processes, we couple reactions. We do a reaction that releases energy to provide the energy to do a second reaction that requires it. And the big gear is supposed to be the exergonic or energy releasing reaction, and the small gear showing that a portion of that energy that was released is used to drive the endergonic reaction. And the relative size of the gears there is kind of important if you want to think about this metaphor. And the relative sizes are important because there's always some energy wasted when we are doing energy transferring or energy transforming reactions. Some of the energy is going to be lost as heat, and while a mammal like us can use some of that heat for sort of a valuable reason, to keep our bodies warm, to keep our metabolism humming along at a rapid rate, it's still something of a waste. The heat can't be used to do meaningful work, so there's always going to be a little loss of energy. ADP and ATP are things that most of you have probably heard of. ATP is the energy molecule, the sort of energy currency that living things use to drive lots and lots of cellular processes. We make ATP by connecting an adenosine diphosphate to a third phosphate group to create ATP. And ATP is made during the processes of cell respiration and during photosynthesis, and is used as really the fuel to drive a lot of the chemical reactions and chemical processes that take place in cells. And giving up that third phosphate is like transferring energy. So when we talk about using ATP in a cell, using it for energy, what we're talking about is that an ATP molecule will donate, will transfer its terminal phosphate, its third phosphate in that chain, the one that's circled there. It'll transfer that to a different molecule, and in doing so, transfer some energy in the process. And it's that energy that helps to drive a lot of processes inside cells. And the last slide is just showing two of the coenzymes that are critical in the process of cell respiration. These are the electron transporting coenzymes NAD and FAD. That's nicotinamide adenine dinucleotide. It's no wonder we call it NAD. And flavin adenine dinucleotide. No wonder we call it FAD. These are the electron carriers that can help the enzymes in our respiratory pathways take away high energy electrons from food molecules like glucose and use them to help drive the production of ATP. It's nice if you're familiar with it, NADH and FADH2, but I'm certainly not going to ask you any details about its structure or exactly how it works. These are the molecules that do electron transfers during respiration. We're all done.